So now actually majority of us actually we will order food online, and then they will come with all these plastic container, right? So definitely this kind of packaging, plastic packaging actually will increase the microplastic, okay, through the earth. Very soon you can see, okay, this is another problem, okay, to the earth. So this plastic waste can be degraded into microplastic by the exposure to the UV light, and then the posting hidden challenges to the marine animals, humans, and environment. So of course, it actually takes years, okay, to degrade the plastic into microplastic, but you can see the amount, right, that actually generated by human, right? All this actually because of the human being. All right. Microplastic pollution is also a serious problem in the UK, as they have exported around 4.15 million tons of plastic packaging from 2010 to 2017. So if you look at Malaysia, right, just like what I mentioned, from last year until today, okay, due to the pandemic, right, we actually noticed that the consumptions of plastic packaging actually is increasing, right, because of the online food ordering. Okay, so and then if according to the statistics, right, so Malaysia actually ranked as one of the highest country that actually have this higher consumptions of plastic, right, among the Asian country. So why do we study microplastic? And do you know that microplastic pollution is one of the major environmental problems that affect the society today? So by estimations, based on the global average of microplastic injections, it can be as high as 5 grams per week per person. So definitely if you look at the amount, right, so 5 grams actually is not a high amount. But if you look at the accumulations, so definitely this one is not a short-term effect, but you can see possible actually you may see the effect, okay, in a long term. All right. What is microplastic? You actually can find this microplastic everywhere, right? So even though from your mineral bottles, right? So definitely you can see they have the very minor, tiny microplastics, right? And even though you look at some detergents, okay, the shampoos, okay, or actually even though like body wash, right? So they actually contain this microplastic. And this is actually plastic particles. Their size actually is not very small. If you really because the size actually you can see. Okay, you use your naked eyes. The size is about 5 mm, which they have a different sizes, shape, and also their chemical compositions. They have a noise, okay, it's quite noisy. I'm not sure whether, okay, you all still can hear me, okay, I will just continue. All right, So PVC actually is made of vinyl chloride monomer and has been widely used for productions of like the materials that they commonly use in the construction site. So you look at the buildings materials. So many actually is PVC materials, and even though you look at the toys and the food packaging materials, right? So all these actually is PVC material. Okay, so construction site actually is the industry that can produce a lot of PVC waste. And then definitely, okay, they are very useful because like your piping, everything you do need to have a PVC. But then, okay, some of the waste, okay, if you have a chance, go to the construction site, you will realize, okay, they just throw the PVC. So how they do it, right? So they just dig a hole, and then, okay, they just dump it, and then they throw it, okay. So definitely, this is not the proper way to discard, okay, this PVC. And definitely, it's actually take years, okay, to degrade, okay, but they are not fully degraded, okay. You still manage to see the nano, the microplastic, okay, from the soil, from the water, and from the ocean. So this one actually cannot be recycled. Next, PVC is also added with some plasticides such as phthalate and BPA. So which these actually will increase the degradation times of plastic. So you can see now they come up with a lot of ideas, right? So to design a replacement, okay, they can replace PVC, but definitely that one is very hard and difficult. So this kind of residue actually can cause genotoxic damage to us. For example, like the PPA, right? They will cause lipid metabolism disturbance and oxidative stress, and which actually have a very high risk of getting chronic health effect. For example, like diabetes, heart disease, obesity. So this kind of chronic diseases, right, actually is not developed 1 or 2 days, right? So this is actually due to the accumulations of the DNA damage. And one of the possibility is because of the contributions of these so-called like the microplastic, right? Because microplastic actually, it won't dissolve, right? When it's actually through your food consumptions, we can't dissolve it, right? But they will have some indirect effect or impact to human health in the long term. Currently, there is a limited evidence claiming that microplastic consumptions have any direct impact on human health. In recent years, actually, they have many research or publications about this microplastic. So most of the work, they actually, for example, they identified microplastics from marine organisms, and also they identify or characterize the types of microplastics from the bottle, right? And also different food container. It's not difficult to get the publications, right? But then if you look at the effect or impact of this microplastic, how it's actually give a direct impact to human health, actually is quite limited. There are some studies conducted on animals confirm that microplastic could cause harm physically and chemically. For instance, small animals might experience blockage in their feeding appendages, and they will cause some pseudo sensation. And this potential harmful effect from microplastic particles and associated chemicals towards human health should be further investigated. So we cannot just stop. That's why actually we have to do more convincing scientific data to prove that it really show some acute effect, or even though like the chronic effect, right? So therefore, an initial in our team, okay, they have some initial work have been done. We actually purposely choose the PVC microplastic, right? Because like the surrounding earth actually they got a lot of constructions. Because the town that I in now actually is developing, right? So we easy to get a very means of the constructions, right? And I believe that okay, these are PVC microplastics. Also they will go underground, means actually like for example underground water, right? So how is actually is affect to us, okay, through the pipe water. So initial work PVC microplastic induced oxidative stress and pro-inflammatory responses in ICR mice was carried out. All right. To date, many microplastic related studies have been conducted, especially in the histopathology part. But there is still necessary to study the effect of PVC microplastic effect in mice. Why actually we choose to use mice? Okay, because this mice model actually is more simple, and then definitely it's more convenient for us to have this marine model, right, using the mice sample. So according to the fundamental study, microplastic actually show negative effect to metabolic system. Because we really done some systematic review, okay, before we restart. And this work actually is quite new, okay, in our lab. The mRNA expressions of genes involved in the metabolic system, actually they got many genes. We choose the genes actually because they are well known, is important in the metabolite system. So one actually is a PPAR gamma and CD36. So this one we use. And moreover, okay, besides that, okay, because of these two genes, right, is how to say, they are more common, okay, for the oxidative stress study, right? That's why they were chosen, right? And furthermore, why actually choose a PVC? Because there's still lack of the study on the specific microplastic effect to mice. The model we're actually using the mice. How about the pro-inflammatory responses? Previous study proved that microplastic have negative impact on the immune system. There are many genes involved in immune systems. However, only two genes were studied, right? So we only choose the TRAF3 and FKB1 genes, and both genes are involved in the pro-inflammatory events. When we induce a high microplastic concentrations, so try to imagine when you actually take in high consumptions of microplastic daily, right? So definitely it will trigger this oxidative stress and increase mutation frequency. As we know, once mutation frequencies is increased, then it actually will cause the chromosomal aberrations in cell. Especially, okay, it may cause like abnormality during the cell cycle, right? And chromosome aberrations in cell, right? So during the cell cycle, it may cause an aneuploidy. You can see they have an abnormal number chromosome in the cell. So this actually is the main reason why it's caused means an unstable of genome. Right, so definitely, okay, when they have an high aneuploidy in the host, right? So definitely the risk of getting cancer is also very high, right? So when we found that, okay, these oxidative stress, and it show when they have an increasing of the mutations frequency, then another way is to double check, okay, whether they show any anomaly beside of the chromosome aberrations, right? You will find that the gene expressions actually is extremely different when compared with a normal. So we actually did this, okay, through the animal study, right? It's actually really show us, they show variations in the gene expressions when we compare with control. This was actually carried by my final year project students, and they actually have a different group. So one group actually study on the gene expression part, and concurrently actually another group is doing the histopathology. So I did not include the histopathology part, okay, in today's presentations. So I just focus on the gene expression part, okay, using the qPCR. So for this work, the student actually requested to investigate the association between body weight of mice and after treated with PVC. So they actually do comparisons, okay, whether they have any dissimilarity of the body weight, okay, before and after PVC treatment. And after that, okay, we'll try to compare the mRNA expressions of the PPAR gamma, CD36, TRAF3, and NFKB1 for the treated and untreated mice using the qPCR. And lastly is to investigate the alterations of the expressions of the genes after the consumptions of the PVC microplastic. Okay, so microplastic exposure to human, definitely yes, because like you exposed to this microplastic daily. I believe that you notice it, but you always hope that actually it will not cause any harmful effect to your health. Just like I mentioned, this is a long-term effect, it's not a short-term effect. According to the study, 83% of tap water samples around the world already contaminated by microplastic, which actually we did not, if you really drink water directly from the tap water, definitely you drink along with all these microplastic. And I believe that, okay, now actually with a high technology, they got a lot of like water filter, right? So all this type of systems, filtering system, actually they can help you to reduce the microplastics consumption. Besides that, okay, we also noticed that the microplastic contaminations are found in honey, beer, and sugar, right? Because why actually they have these microplastic residues? Because of the container, right? So like the sugar and honey, right? All these actually they need water, right? So definitely you might able to see and you may not able to differentiate, right? Okay. So this is another way that why actually you take in the microplastics as well while you're eating. Microplastic can be transferred from fish to high-level marine organisms, suggesting traffic transfer can be a potential key pathway of microplastic exposure in species consuming the whole prey, such as human feeding on seafood and fish. In the Chinese populations and in Malaysians, they actually take in a lot of seafood, right? So like fish, right? So we actually like a lot of seafood, right? So definitely the previous study have shown that, okay, they got very high amount of microplastic from marine organisms. Then, of course, we actually clean it properly, okay, before we cook. Microplastic is exposing to humans in our daily life, and then it's actually increasing concern on the potential effect caused by microplastic injections on animals and humans. All right, you look at this slide, which actually contains the different research area. Right, for example, like immune activity, oxidative stress, and the fish swimming behavior, and also the lipid energy metabolisms, intestinal damages, gut microbiota, and the body mass, neurotoxicity, genotoxicity. All these kind of research activity have been carried out before and after the contaminations of microplastic. They actually have done a lot of work, right? So they look at the pathological effect of the microplastic, okay, in the respective organs and also the system. In this study, so although there have been previous study or many study, right, that have been done on the effect to evaluate the effects of microplastic to immune system. However, most of the study focus on histopathology study. So if you really go through this kind of articles, right, so actually it's not difficult to get a histopathology study. Is it when compared to the genetics study, right? Why actually I study genetics level, okay, because like you may able to see the minor changes, okay, based on the gene expression, but you may not able to see the minor changes based on the histopathology. So yeah, that is the part that conducted by my group, right? Of course, the histology part is conducting by my colleague. So besides, majority of the study involved marine organism instead of using the mammals. This is also like of a study on both TRAF3 and NFKB1 genes. Of course, for the PPAR gamma and also the CD36 have been studied previously, right? So because we actually want to see any significant difference, okay, between our study and also the study that done by other group. So definitely this TRAF3 and NFKB1 genes, right, is so far is lack of the study, okay, for both genes. So definitely this is something I think is additional, okay, for the microplastic study. Okay, so just like I mentioned, the effect of microplastic that have been done previously, okay, especially in the hepatic lipid metabolisms. So how it's actually give an impact to the hepatic lipid metabolisms. So look at these two research activities they carry out by two different research groups. One actually is using mice sample, and the other one is using the zebrafish. So if you look at when for the mice sample, okay, they try to treat the mice sample with 0.5 and 5 micrometer of the PS microplastic, right? Is induce the expressions of PPAR gamma and CD36. But for the zebrafish, okay, they use 5 micrometer PS microplastic, and it show, okay, the PPAR gamma down-regulated, right? So this is something show differences, okay, when they actually in a different organisms. So most probably, okay, because of the behavior of the organisms is different. All right, so now we move on to the slide. Okay, this is about the informations for the genes of interest. PPAR gamma is located at P arms of chromosome 3, regions 25, band 2. This is a nuclear hormone receptor which controls the gene expression involved in lipid homeostasis. The next gene, so this gene is CD36, right, which is located at Q arm of chromosome 7 at region 11 and band 2. So CD36 is a member of the class B serology receptor family. It actually is a receptor for ligands, which is oxidize a low-density lipoprotein long-chain fatty acid, which also facilitate transport of fatty acid into cell. From an articles, they actually show that the 3D CD36 expression is regulated by PPAR gamma. Right? Next slide. Okay, next slide we actually talk about the associations of PPAR gamma and CD36. So if you look at this diagram, so it actually shows the associations of two genes, of both genes. So in the absence of ligands, if you look at the diagram, in the absence of ligands, the PPAR gamma will form a heterodimer with retinol X receptor alpha and bind to PPRE. This is associated with co-repressor that turn off the gene transcription. Once activated by ligands, the co-repressor will be replaced by co-activated, then triggers gene expressions. Upregulations of PPAR gamma also will lead to upregulations of CD36, and thus it increase the fatty acids uptake and increase the triglyceride accumulation. All right, now we discuss about the genes that involve in this inflammation response, TRAF3. This gene located on chromosome 14, Q-arm regions 32 and band 32. So its player functions is encoded for TRAF3 protein that mediated signal transductions from members of TNFR superfamily. It is Okay, next. Okay, the next one is NFKB1. So NFKB1 encode a P105 protein, which have been cleaved to produce a subunit of transcription factor NFKB. So this gene is located at the Q arm of chromosome 4 at regions 24. So look at what is these are TRAF3 and NFKB1. So both genes are involved in the NFKB signaling pathway, which is divided into the classical and alternative pathways. So what you can see from this diagram is like NFKB1 genes is involved in the classical way, while TRAF3 is involved in the alternative pathway. Right, so which you can see I highlighted them in red color. Right, so in the classical NFKB signaling pathway, So during the activation stage, it will increase recruitment of TRAF2 and IKK1, which will induce TRAF2 mediated NF-κB1 activations and thus cause nuclear translocations of NF-κB. While when it's in alternative NF-κB signaling pathway, during the activations, when it's activated, it will cause recruitment and target degradations of TRAF3 and induce NIK-mediated NF-κB2 activations. Then it will cause nuclear translocations of NF-κB. Okay, after introducing you about all the genes and some background about the genes and the previous study that have been done on microplastic towards organisms. So maybe I will just discuss some of the material that have been carried out to achieve the objective of this work. So the material that we use is we choose the ICR strain. So this ICR strain actually is commonly used, okay, in the in vivo study, and the mouse breeding is carried out in Utah, in my university. So we choose 7 weeks old female mice for this study, with the reason these mice already reached the stage where it's actually show the ability to reproduce and they can grow quickly. And the microplastic that we use, actually this microplastic is the PVC microplastic is prepared by my colleague, right? He actually helped me to prepare it, and then the particle size is about, by average, is about 154 micrometer. And definitely they have a different sizes, vary from 69 to 360 micrometer. And microplastic actually cannot be dissolved, right? But why we actually choose olive oil, because it's more easy as a delivery. So it's easy, okay, for us to transfer it to mice, okay, through the force feeding. And this olive oil actually high in monounsaturated fat, low in saturated fat. According to many statistics, and then also the study have proven that olive oil actually will not cause any side effect to, or they will gain weight, right? So definitely they will not increase the body weight. So from the olive oil. All right, so this one actually is the experiment designs that have been conducted. The design actually, the samples or the mouse have been divided into 4 groups. So we have a control groups, low dose, medium dose, and high dose. So the calculations, okay, like how to calculate this one, actually we calculate based on the assumptions. We take about 5 grams per week. So according to the statistic. So the control actually we just prescribe, okay, this one actually we don't, we prescribe with olive oil, right? And then the low dose contains 0.05 milligram per microliter of PVC microplastic. And then the medium dose consists 0.5 milligram per microliter, and the high dose consists 2.5 milligram per ml, right? So the low and medium dose represent chronic conditions, and then the high dose represent acute condition. So can you imagine, okay, you take in 1 spoon of microplastic, and we want to see, okay, whether they have some acute effect, okay, to human health, right? So definitely we wouldn't do it, right? So this is something like we would test on the animals using the mouse. So the mouse that we use actually is a 7-week-old mouse, right? So which weight is about 21 to 35 grams. Okay, and the calculations of these treatments is given based on the body weight of the mouse. So you can refer to OECD guideline, okay, when you actually do these calculations. All right, look at the methodology part or the designing of the work. So first of all, because we're actually using animal sample, that's why we need to apply ethical approval from Utah. So after getting the approval from Utah, then only we can carry out the work. The microplastic treatments were carried out for 9 weeks, and then after that, the mouse will be sacrificed, right? And then they will further to RNA extractions. So if you look at the microplastic treatments, okay, throughout these 9 weeks, the student actually provide the microplastic through oral gavage. And then it's a force feeding. Then every day they have to record the body weight of the mouse. After 9 weeks of treatments, the mouse actually will dissect and the tissue will be means that the tissue was harvested. And the liver harvesting actually, the liver actually we do need to share, right? Because if you look at the mouse, right? So actually it's very small. Can you imagine the size is just about 21 to 35 grams? And then even though like the liver, the color, the weight, the size, actually we also do some measurement to compare whether they show any significant difference, okay, between the control and also the treated, the PVC treated mouse. And for the RNA extraction, definitely we did not use the in-house RNA extraction buffer. So we actually use the one actually is a commercial, right? It's a commercial kit. When we do mRNA gene expression using qPCR, the quality of RNA actually is important. Right, because like the purity of the RNA actually will affect the result. Because like if your RNA is not pure enough, so definitely your gene expression actually they will come up with a lot of mess, right? From the graph, okay, and even though from the CT value you can see obviously that is a mess or a noise, a background noise. So RNA quality assessment also we use kit, okay, to do the means to purify the RNA. Then we will check, okay, the present bands of RNA, and then double check with their purity, okay, using the nano drop. cDNA synthesis also, we purchase the cDNA synthesis kit, right, to achieve to synthesize the cDNA. Then so on, similar like the qPCR kit, right? Also we use the commercial one. Okay, and lastly, okay, we do comparisons among the 4 groups, okay, based on the gene expression. Okay, so this is some of the mRNA which have not been purified. So definitely the quality improved. So after the purifications. So this actually is the 4 primer pairs sequences that have been used, okay, to carry out the work. Housekeeping genes that were used is 18S rRNA, right? And then definitely if you look at the amplicon size, okay, the amplicon size actually is meet the expectations if you want to carry out the qPCR. Okay, so I want to introduce, okay, the sequence, the amplicons of the TRAF3 and NFKB1. So NFKB1s, okay, the amplifications regions actually fell into these prominence regions. So this region actually is a prominent component of the programmed cell death or apoptosis pathway, which are found in a number of other signaling pathways, including those that impact inner immunity, inflammations, differentiation, and cancer. And while for the TRAF3, so the molecule will serve as adapter proteins that link to TNFR and downstream kinase cascades, resulting in the activations of transcription factors and regulating of cell survival, proliferation, and stress responses. So this actually is why I want to introduce these two, because these two genes actually is the information is quite limited. Okay, if you want to refer to the microplastic work, but definitely for PPAR gamma and CD36, it's not difficult because like you can get the relevant articles. So this is a PCR, qPCR amplification setting, right? So it's just a very symbols, right? So we actually set these qPCR setting based on the manufacturer guideline. Then definitely, of course, we did the students have done some optimizations to their entering temperature. So in order to get a more standard, the optimum or the best conditions. All right, so now move on to the observations and the findings of this project. So definitely we have achieved all the objective. And if you look at these tables, okay, this table actually show the total weight increment comparing the mouse, okay, in from that 4 groups on control group, low dose, medium dose, and high dose. So after conducting the statistical analysis, it do show no significant difference among these 4 groups, and no much differences between control and treatment groups. And high dosage group mice exhibit a greater weight gain compared to low dose and medium dose. But when we do the statistic analysis, they show no significant difference. But it actually do show slightly higher compared with the low dose and medium dose. No significant difference was observed in the weight increments between high dose group and control. And then from this study, what we can say, it does not show any pseudo sensation during the treatments. So the feeding activity was not altered by the PVC microplastic consumption. Okay, if you look at the clear observations, okay, from week 0, from the day 1 until the treatments after 9 weeks, okay, throughout these 9 weeks, right, you can see the changes, okay, the increments of the body weight. Okay, so when you look into this detailed trend of weekly weight increment, there was a decrease in weight gain, followed by weight loss in all mouse. These conditions occur during the 2nd and 3rd week. Can you see the differences in the 1st week, 2nd week, 3rd week? Right, you can see they have some differences, right? In the control, low dose, medium dose, and high dose. So this is actually some record that have been done. So as for the medium dose, okay, the mouse loosen their weight from week 5th and 6th. The reason why the mouse, they actually take in, they have a less food consumptions, at the beginning of the treatment. It probably is due to the stressful oral gavage procedure because they are not yet adapt to the environment change, right? Because of the stress, because all this actually is due to the forced feeding. So they regain their appetite on the following weeks due to the adaptations, okay, to this forced feeding. So after that, we can see in week 8 and 9 actually means that you can see they did not show any inconsistent of the increments based on their weight management. Okay, we look at the some possible explanations, right? So look at the changes from week 0 until week 9. So this can be due to the complications of oral gavage that cause mice to be stressful and reduce the food intake. However, the weight gain of mice increase again after the mentioned significant decrement. The possible reasons of this can be the mouse have adapted to the changes, as supported by the finding which they show that mice actually will less stressful after than the 8 consecutive days of force feeding as compared to early phase. So they show significant decrements in mice weight gained at 3rd and 6th week of microplastic treatment. So this one actually is during the process. So another beside of this reason, another possible reason because of the activity is carried out by different person, because the student actually taking turn to take care of the mice, right? So possible this is another error, another variation that caused by human being. The possible reasons of that could be due to another one. If you look at the body weight, okay, the measurements, right? So besides of a human's error, okay, it could be due to the size of the concentrations of the microplastic was insufficient to trigger weight impairment. Unless actually you have a very specific size, right, that you can make a comparisons, like you're comparing whether which size actually may have a sufficient that trigger the weight impairment. Therefore, this can be implied that PVC microplastic exposure did not cause any reductions in appetite of mouse from our study. And then you can see that does not lead to any decreased food intake activity. All right, so look at the morphology of the mouse liver. Actually, we are not just study on liver, right? So other parts of the organs also we try to investigate the differences. So I only show you the morphology and then the gene expressions from liver. You can see, okay, they have no remarkable difference on size measurements, and no changes in color intensity, and no hypertrophy or atrophy. But then when you look at their gene expression, you can see they are slightly different, and then they show some differences. All right, so now I move on. Okay, you look at this is the transcriptional levels of PPAR gamma in mouse liver. So I'm not sure whether it's quite small for you, right? So you can see this one is a control group. The blue color is a control group, and then okay, the orange color is a low dose, and gray color is a medium dose, and the yellow color actually is which is in high dose. So the PVC microplastic effect on PPAR gamma was evaluated, okay, based on these 4 groups. So control groups was set as control. So when they calculate, okay, the expression level, the control group actually will be used to normalize, okay, the expressions. So for medium group, okay, if you look at the medium dose, there was no significant different change, right? If you read, even though you look at the comparisons, right, there actually no significant difference, okay, comparing with the control. But for low and high dose, the gene expression increased by 2 and 3-fold changes, respectively. So under normal condition, the PPAR expression in liver is low. The upregulations in this study could be an acute response to microplastic exposure, indicating that triglyceride accumulations in liver may have increased. So of course, we do need to have a further test, okay, to prove it, okay, by measure, okay, the level of triglyceride. Okay, next we look at the changes, the transcriptional level changes of CD36 in mice liver. So when you're comparing the 4 different groups, both low and medium dose group, they show down regulations expression, right? When it's actually compared with the control, it show low decrease the expressions of CD36. And normally CD36 has low basal liver expression, but it could be upregulated when there is a fatty liver disease or obesity, right? So this actually is a common scenario. So when it's actually have a fatty liver disease or obesity, they may have a high level of CD36. However, an average decrement trend suggests that the PVC microplastic induce an acute response by decreasing fatty acid uptake and increasing non-esterified fatty acid plasma level. Okay, next that I would like to discuss. Okay, this one is to compare the PPAR gamma and CD36 mRNA expression level. So we want to see the interactions of the expression level, right? So by making a comparison in changes in both gene expression, right? We can see obviously we can make a conclusion that PVC microplastic induce upregulations of PPAR gamma, but diminishes CD36 expressions. As we know, okay, after get the supporting articles, we noticed that this PPAR gamma actually is more susceptible to be affected as compared to CD36. The expression is different among different concentrations. So we've impaired that they may be caused by uncertainty like the size of particles, the amount of plasticizer like BPA, DEHP, and that's why further investigations need to be carried out to confirm the result. The possible reasons for the variations in both gene expression might due to the effect of plastic additive. So the plastic additive leach out from PVC, such as like BPA, DEHP. So this actually may alter the gene expressions. So according to the previous study, both BPA and DEHP increased PPAR gamma, but decrease CD36 expression in the treated mouse. Okay, next. Effect of PVC microplastic in hepatic system of mouse, right? So against the plastic additive may cause decreased breakdown of lipids and increased triglyceride build-up in liver, and thus is cause hepatic lipid metabolism disturbance. So both BPL gamma and CD36 is important in lipid metabolisms. So you can see it, okay, when you're comparing with the non-treated and treated mouse with microplastic or with the PVC microplastic. In additional result from histopathological study done by my colleague, they show infiltrations and neutrophil in liver of high-dose mouse. So this observation suggesting that inflammations had occurred. And this is consistent with our result, which demonstrated an upregulation of PPAR gamma in low and high-dose group, and thus suggesting an effect of DEHP that disturbed the antioxidants balance in liver cell and induced oxidative stress in mice. So we suspect, okay, because of the PVC is coated with DEHP, right? So we suspect it could be due to the DEHP, right, that disturbed the antioxidants balance in liver cell. All right, so you look at this slide, okay, this slide shows the possible reasons for less notable effect in low and medium dose group. So as most of the discussed effect are seen only in high dose, because a high dose actually is acute effect. There are some possible reasons for the less notable effect in low and medium dose group. First, the mechanisms of actions of endocrine disruptor are complicated and possibly epigenetics. Therefore, any low and medium dose might result only in long-term changes in gene expression. And this treatment actually only prescribed for 9 weeks. So we may not able to see the significant change based on their gene expression. So we predict that, okay, you may able to see the result long term. So which help the disease develop. Just for example, like the chronic diseases and also the cancer, right? Secondly, microplastic can cause DNA damage that result in mutation, loss in heterozygosity and chromosomal rearrangement. That is the possible will cause the genomic instability and aneuploidy, okay, because of the accumulations of the microplastic will trigger the DNA mutations. Therefore, despite the dose of microplastic treated to mice may efficient to trigger an acute response and causes DNA damage in mice. However, observable or physiological effect may not be seen in short term. Okay, after complete, okay, the discussions on antioxidants related genes, okay, now we look at the genes that play important role in the pro-inflammatory responses. The TRAF3 and NF-κB1. Look at the graph, right? So we actually compile, okay, the TRAF3 and NF-κB1 in one graph. So because like both of them, they show a positive correlation. This graph shows the comparisons of the genes. So both genes were down-regulated. Okay, if you're comparing like the control, okay, comparing with low and medium, right? So both gene expressions were down-regulated, right, in the chronic stage, okay, in low and medium dose. So, but the high dosage group, okay, you can see they're extremely different, right? It was up-regulated. All right. So we can see, okay, the PVC microplastic really alter immune functions in chronic microplastic treated mouse. So the low-medium dosage group represent the chronic microplastic treatments. This design actually is just a model to represent the actual conditions in humans, right? So that's why the low and medium dosage group, they will represent the chronic microplastic treatments. And results show that down-regulations of both genes in these two groups. Therefore, this is postulated that the low concentrations of PVC microplastic is insufficient to cause inflammatory event in mice, but can cause alterations in the immune system. This is because when both genes were down-regulated, there will be high expressions of TNF-alpha. Interleukin-6 and interleukin-10 will alter the cytokine balance in the immune systems. The Treg cells will also increase significantly, and subsequently it will inhibit T cell proliferations and cytokine productions. This might be telling us that even though there is no definite indication to show signs of inflammations, but the low concentrations of microplastic intake for a period of time could have affected the normal functions of the immune systems. All right, so now we move on to the high dose group, which actually represent the acute conditions and how it's actually show the variations in the transcriptional levels for the genes. So these are high concentrations of PVC microplastic may have triggered the CD44 or CD4D mediated signaling pathway and lead to detectable inflammations in the mice liver. Okay, if you look at both genes, okay, the both genes, okay, that we use, okay, for study the inflammations, right? They actually have demonstrated that positive correlations with the CD4D mediated signaling pathway. So when the CD4D ligands bind to CD4D, there will be recruitment of a signaling complex consisting the protein TRAF2 and TRAF3. That is the step that causes the upregulations of TRAF3 genes. Then autoubiquitinations of TRAF2 will activate the classical NF-κB pathway. And this is the step when NF-κB1 genes will be upregulated. And this will lead to NF-κB target gene expression. The targeted degradative ubiquitinations of TRF3 will activate the MAPK pathway and thus lead to an MAPK target genes expressions. The activations of transcription factors NF-κB and protein kinase MAPK lead to a series of pro-inflammatory immune responses. It is saying so because if you look at the result, the observed result, NF-κB activation gives rise to inner immunity, inflammations, and anti-apoptosis. There were studies mentioned that NF-κB activation is highly associated with carcinogenesis. As for NAPK, signaling, it regulates cell growth, proliferations, and differentiations of immune cells. The result of this particular high-dose group are tally with finding of histopathology result from other groups that show the presence of eutrophic infiltrations in livers of mice treated with high dosage of PVC microplastic. Microplastic may cause aneuploidy in cell. So other than that, the upregulations of TRAF3 and NF-κB1 genes it could be due to the aneuploidy in cell, which actually caused by the consumptions of PVC microplastic. The high concentrations of this PVC microplastic can cause oxidative stress that can induce mutation in the gene and then causing chromosomal aberrations in the cell. This will lead to aneuploidy in the cell and thus causing the genes to express higher than normal. So this one actually is based on the expectations, right? Because like this kind of phenomenon is very common, okay, found in the cell cycles, right? And how it's actually caused the tumorigenesis and then the carcinogenesis. Now it's clear that PVC microplastic can cause inflammations and alterations to the immune system functions. But how do they trigger the immune system? There are two main reasons. The first one is oxidative stress. So the journal or other articles also stated that MP or microplastic exposure has caused oxidative stress in mice and lead to the activations of MAPK signaling pathway. It happens because of the MP can release reactive oxygens or ROS species that oxidize lipids and proteins. And this can cause cell destruction and which will promote inflammation. So besides trigger oxidative stress, okay, now we look at the plasticizer. The second way is through the presence of the plasticizer in PVC microplastic. So this plasticizer, right? So normally, actually, the purpose is used to coat as a agents, right? That they put these plastics. The two main plasticizers that we found, right? One is this phthalate and then BPA. Phthalate can cause cytokine imbalance. As for the BPA, it will disrupt the cytokine P450 enzymes in hepatocyte and further lead to oxidative stress. So besides that, okay, BPA also they contain the peptide that act as an immunogen. Okay, so look at some of the from the students' work and from these current observations or the findings. They have some proposal for the future study. Okay, what actually we should look into, right? So as we noticed that, okay, the plastic sizes is one of the important components because if we look at the size of the weight, okay, of the mouse, right, and they are not significant change, right? So we want to see, okay, how about when they only consist the plastic sizes only, okay, or these are chemical residues, right? So possible we can consider try to treat the mouse with BPA only, right, or try to treat the mouse with DEPH, right, to evaluate how about the gene expression, because we still uncertain, okay, if the size of the PVC microplastic actually will give some direct impact to the gene expressions or not. And then also we want to look at just the BPA only, okay, where they show any significant difference, okay, based on their gene expression. Another one actually is possible we can increase a larger sample size, okay, because so far this study carried out with only 4 mice, okay, in one group. So if a larger sample size, definitely we can have more treatments, right? Comparisons. For example, they have a group that just treated with PPA, and they have a group just treated with the particular specific size of the microplastics, right? So that we can have a wider, okay, comparisons, right? Okay, based on the gene expression level. And then you may consider the different types of microplastics, right? So want to see, okay, are there really significant difference, right? And this one actually is based on the understanding level, right, for us. We want to see whether they show any variations or dissimilar, okay, when they consist of different types of microplastics. Another topic actually is quite interesting that we are going to study is analyze the gut microbiota, right? So as we know that this gut microbiota, right, so it's actually linked to is really very show associations with all these nutritions, okay, even though like the nutrigenomic study and also our health, right? So they actually is correlate. So possible actually we can look into this microbiota, okay, the gut microbiota, and see what is associations, okay, and then as we know, it's a toxicity targets for microplastic, right? And then to investigate the interactions between PVC, microplastic, and then the microorganism as well in gut microbiota. I actually they have publications that have been done. Okay, they look at the diversity of the microbiota. All right, so before and after treated with microplastic, right? They want to see whether actually they will affect the positive, means the beneficial microorganisms, okay, in gut. So this one actually is a very interesting work, right? So definitely possible, okay, is may actually help us to understand more about the human health. But the issue here is difference, okay, in human actually they have a difference of practicing of a diet, or even though like the culture of their food dietary, right? So then possible their gut microbiota actually will show some differences, right? So this is another part that we can study, right? So which is more related to microplastic, the associations of microplastic, and then the nutritions, okay. Okay, conclusions. Okay, just a very quick. Okay, I'm already come to the conclusion part. So in conclusion, right? Overall, what we can see, this study, okay, the TRAF3 and NFKB1, they really show the positive correlations of mRNA expressions in the mice treated with different dosage of PVC microplastic. And this study also showed that the injections of PVC microplastic have caused alterations in the immune system function in mice when you actually comparing with the low dose, medium dose, and high dose, right? And the inflammations in the mice treated with high dose of microplastic, actually you can see they really show over expressions. Microplastic, this PVC microplastic does cause significant change to the body weight of the mice. So as the mouse is a common mammals model, hence it is predictable that the health risk of microplastic to animal could be reflected to human as well. So we hopefully that the result actually is convincing and then could be suitable to reflect the humans' health conditions. As we actually will take in microplastic through food daily in our daily life. All right, so so this one have been explained just now, right? Okay, and last but not least, I would like to acknowledge my team members, Dr. Uy, who actually is from the Department of Chemical Science, and then he is the person that prepare all these PVC microplastic samples, and also Dr. Dan, for his knowledge, doing the mouse breeding, and then he also guide the students, how to conduct this type of oral garbage, right? So the force feeding, to mouse. And then in order to reduce the stress to the mouth, to the mouse, because handling actually is one important technical error, right? They may cause a biased result in their body weight, and then of course also they may have some uncertainty during the procedure, right? If you do not have proper handling. And then I also would like to thank Dr. Michelle, right? So she is our group members who is focused in the histopathological study, right? And so that the gene expressions can tally with the observations in histology part. Then definitely, some of the histopathology work actually is not significant difference when they try to comparing with the control low dose, medium dose, and high dose. Means that when they're comparing with the chronic condition and then acute conditions, you may not be able to see significant difference, right? But then you can see a slight difference, based on your expression level, right? So because these PVC treatments actually may cause they may cause abnormality during the cell cycle, right? So it actually may cause DNA damage, and then indirectly actually it will affect the DNA repair system as well. And I would like to thanks for my girls. This is final year project students. They really have done a very good job conducting all these project activities, and they really is very dedicated, right? So they actually take care. They do the mouse breeding, from the early and until they're completing all the project activity. right? And lastly, I would like to thank Utah, okay, because of the fundings and also the lab facility support. Okay, so thank you very much. Okay, all this is my presentations. Okay, my slide presentations.
Thank you, ma'am. The session is now open for discussion.
To a receptor, ma'am.
May I know is it animal studies is carried out in your university?
Any other questions? Or you can post your questions in the chat box.
Taking care of animals actually is tedious work. So that's why throughout the studies, okay, for master students, of course, they are full times, okay, because they are in Utah research activity. We have a postgraduate programs. So our postgraduate program actually is a full-time research. And for the final year students, okay, while they're conducting their research activity, they do need to sit for their examination for other courses. Yeah, so that's why, okay, this work, okay, this kind of work or quality of work which is done by the final project, I would say that, yeah, it is they are really okay have spent a lot of effort, okay, to complete it, right? So without them, actually, I cannot finish. I cannot do this kind of. I couldn't have this fundamental data for me to continue. Okay, the microplastic work, because so far, as I know, microplastic, yes, they did. I have a colleague from other faculty. Okay, like the green technology engineering, they really carry out some work on microplastic. So their work is slightly different because our departments actually will focus. I'm focused on health, means the health related topics. But for them, okay, they are more focused on the environmental, right? So like green technology, right? So they want to see what is the effect of this, the type of microplastic, right? What type of impact, okay, to the environment. So they really do characterizations and collecting the microplastics from different regions, from soil, from underwater, river water, right, and also from ocean. So they're actually comparing the distributions of this microplastic. So that's why actually I really lack of this fundamental data. That's why this initial work was carried out. Okay, I have one question. Is that size variation? Where I can see the questions. The variations of the microplastic size, right? Definitely, we believe that they will have a different impact to the extraction level. So in future, we do need to characterize, because like if according to my presentation just now, right? So I do mention that the average size for the microplastic is 150 something, right? But actually, okay, the sizes, okay, based on the analysis, right? So it do show that the size actually from 60 micrometer until, okay, 300 micrometer. So they have a small particle, they have a huge particle. So when they actually do the preparations, so definitely, okay, we just based on the average. We are not based on the specific size, okay, of the microplastic. I would say that it's impossible to remove the microplastic, okay, from water, right? So unless actually you have the specific filter, okay, then don't take outside food, okay. And even though when you actually taking a raw material, right? So like Malaysian actually they would take raw material, for example, in a Malay populations, right? They after clean, okay, the vegetable, okay, they actually will eat raw, right? So just like the salad, okay. So definitely, okay, even though your water bottle, right, mineral water, so all is actually contain microplastic. So which you may not see, because some of them the sizes is quite small, right? You may not see clear, okay, using your naked eyes. So, but of course, we want to see how. That's why they have a water filtering system nowadays, right? So you can see many commercial company, right? So they design this kind of wastewater treatments, okay, to reduce, okay, as much as possible all these contaminants. Right. And of course, in future, we will try to carry out the work, okay, in a fish, right, or from like the seawater, right. So we want to see, like for example, whether actually they have a correlation with the microorganisms, right? Because like if you look at the seafood, okay, organisms, right? So they actually taking this kind of microplastic, and the microplastic actually surrounded, okay, it was surrounded by different type of microorganisms. And they have one study that have been done by NUS, right? So the National University of Singapore, right? They found that, okay, those that cause negative impact to the human health is because of the microorganisms, not because of the microplastic. So that's why actually we want to see just a microplastic, the pure microplastic without microorganism. And they see, we want to evaluate whether they show any significant difference, right? From just you collect a sample from wild environment, right, which already consume the microplastic, right? So if you collect the microplastic from the wild environment, right, it actually contain microorganisms, right? So the result actually is the impact of the microorganisms, not because of the microplastic, right? Actually, got a lot of potential project can be carried out, okay, in microplastic. Even though like you know, they have a UK, okay, UK actually they give a lot of funding, okay, for this kind of work, right? So I believe that even though in India also they're quite focused on this kind of green technologies, right? So this is a very significant studies. Any other questions, okay, from the floor?
I think no more questions, ma'am. Thank you, ma'am. Thank you very much for accepting our invitation. Actually, I should thank Dr. Sainu Vasen for introducing you to us. Thank you. Thank you, ma'am. Surya,
Yes, ma'am.
you can proceed.
Okay, ma'am. So, thank you, ma'am. It was really an informative session.
Now it's time to present the rest of the papers. I would like to invite Dr. J. Raja Vrinda and Mrs. K. Sarojini as the chairpersons of the section. Next on line is R. Saranya with the title Cancer Immunotherapy.
Happy midday to everyone. I am R. Saranya, doing second year BSc Biochemistry in TR College of Arts and Science. The topic of my presentation is cancer immunotherapy. I hope my screen is visible and my voice is audible. Now let us see the definition of cancer. Cancer is the uncontrolled proliferation of abnormal cells in the body. As we all knew that cancer is mainly categorized into 4 types. Carcinoma mainly occurs in our skin, sarcoma develop in our bone, leukemia in our blood, and central nervous system cancer mainly occurs in our brain and central nervous system part of our body. Alpha-2 is the first immunotherapeutic drug which was used for cancer immunotherapy. And Jim Allison, who was awarded Nobel Prize in 2018 for immunotherapy. Next slide. Cancer immunotherapy principle. Principle of immunotherapy is actively stimulate the immune system or passively alter the immune system. It is the basic principle of immunotherapy. It is also called, that is, cancer immunotherapy is also called as biologic therapy or biotherapy. Immunotherapy for cancer was first introduced by Rosenberg at National Institute of Health, USA. Cancer immunotherapy mainly boosts the body natural system and fight against the cancer. Now let us see what is about active immunotherapy. Active immunotherapy aims to stimulate one's immune system which are responsible to pathogen, but not it allow all the cells in the body. It is unique in active immunotherapy. Active immunotherapy is otherwise called as immunomemory, and here are some examples include cytokines, etc. Next, let's move on to the topic passive immunotherapy. Passive immunotherapy means transfer of T cells or antibodies and removal of the whole cell. It is not suitable for solid tumors, and its resistance is always due to loss of target antigen. Next, immunotherapy are mainly consist of 4 types: immune checkpoint inhibitors, adaptive cell transfer or T cell therapy, monoclonal antibodies, therapeutic vaccines. Now let us see one by one. What is immunocheckpoint inhibitors? Checkpoints are proteins and regulate responses to foreign cells given by T cell and check throughout the body. It is called immunocheckpoint inhibitors. It also includes 2 types, namely pembrolizumab, otherwise called Keytruda, and nivolumab, otherwise called Opdivo. There are no much side effects in immunocheckpoint inhibitors, but minor side effects include there are common symptoms like fatigue, abdominal pain, headache, etc. Now let us discuss about adaptive cell transfer. It is a type, that is, adaptive cell transfer is a type in which patient uses his or her own T cells to fight against cancer. Adaptive therapies are advanced therapies in adaptive cell transfer is chimeric antigen receptor T cell therapy. 2 CAR therapies are approved by FDA. Side effect. Adaptive cell transfer side effect is hard side effect called neurologic toxicity and cytokine release syndrome. Next topic, monoclonal antibodies. Monoclonal antibody mainly marks the cancer cell, and that's why it easily finds the immune system in which part tumor specific antigen is spotted and deliver toxic substance to the cancer cell. The three main monoclonal antibodies include naked monoclonal antibodies and bispecific monoclonal antibodies and conjugated monoclonal antibody, and it includes common symptoms. These are also included in common symptoms like dizziness, vomiting, headache, etc. Now, cancer vaccines, therapeutic cancer vaccines. Mainly, cancer vaccines recognize cancer cells and the product itself against them, and it destroys the cancer cell. It's all about cancer vaccines. Side effect of immunotherapies. Side effect only result when side effect would cause in our body. Side effect only result of an overaction of immune system, and it occurs even in weeks or months, or if some person it occurs in years. Most susceptible organ to immunotherapy is liver, skin, lungs, kidney, gastrointestinal tract, and endocrine organs. If someone ill at home, and for their safety, the Oncology Nursing Society, that is in short ONS, provides an ONS immunotherapy patient wallet card. Advancement in immunotherapy. Immunotherapy is the first checkpoint inhibitor approved by FDA and allowing the immune system to better recognize and cure and attack of cancer cell and immune reaction against the cancer cell. That is, it is the first therapy used by researchers to develop and fight against the cancer cell. The most common antibodies is naked monoclonal antibody, as it is more effective. As we see, it is a type of monoclonal antibodies. FDA also approved IFN Alta. It is a drug which have been used for cancers like hairy cell leukemia, chronic myelogenous, T-cell lymphoma, kidney cancer, melanoma, Kaposi sarcoma, etc. Thank you for this wonderful opportunity.
What's the difference between active and passive immunotherapy?
Ma'am, active immunotherapy is develop one immune system, but passive immunotherapy is transfer the T cells or antibodies.
Which one is best?
Can get you.
It's 3:47 p.m.
you hear me? Hello?
Yes,
Hello?
we can hear you.
Okay, ma'am. Thank you. Phytochemicals are responsible for medicinal activity of plants. These are non-nutritive chemicals that have protected human from various diseases. In the current study was carried out in the 3 different seaweeds to analyze the preliminary phytochemical screening, estimation of flavonoids and phenols. Materials and methods. Collection of seaweeds. 3 seaweeds, namely Enteromorpha flexuosa, Gracilaria corticata and Sargassum vitae were collected from Manapad. They are dried and powdered. The sample were extracted by the use of solvent ethanol, subjected to qualitative test for identification of phytochemical constituents, alkaloids, turbinoids, steroids, flavonoids, quinones, anthraquinone, phenols, protein, carbohydrates, glycosides, casein using standard procedures. Quantitative analysis of phenols and flavonoids are also carried out. Then carried out the preliminary phytochemical screening determined according to Brenda et al. 1981. Total flavonoids content was determined according to Chang C et al. 2002. Then phenol content was determined according to Marvat M 2009. These are the 3 algae I am selected. So first one is Enteromorpha flexuosa. This is under the category of chlorophyta. Then Gracilaria corticata. This is the red algae under the classification of Rhodophyta. Then Sargassum vitae. This is the brown algae under the classification Phagophyta. Based on the results, table 2 and table 3. Table 1, table 2 and table 3 represents the bioactive compounds for 3 different species of seaweeds. In my work, 4 different solvents were used for the identification of bioactive compounds. They are petroleum ether, benzene, ethanol and water. All the bioactive compounds are present only in the ethanol solvent. So ethanol solvent is a effective one. Next, the chart shows the total phenol and flavonoid contents of 3 seaweeds. For 3 seaweeds, in the current study indicated that the maximum phenol content was recorded in the green algae, Enteromorpha flexuosa, followed by the red and brown algae. The maximum flavonoids content was shown in the brown algae, Sargassum vitae, and closely followed by green and red algae. Blue color indicate the flavonoids and red color indicate the phenols. Then conclusion. Different extraction seaweeds such as Enteromorpha flexuosa, Gracilaria corticata and Sargassum vitae were found to be active in this investigation. Alkaloids, turbinoids, steroids, flavonoids, quinones, anthraquinones, phenols, proteins, sugars, glycosides and casein among the compounds found in the seaweeds. Extraction solvents have an impact on overall phenols and flavonoids yield. Since the phytochemical cure disease without causing any harm to human beings, these can also be considered as man-friendly medicine. Hence, the ethanolic extract of seaweeds shows many components and may be used for pharmaceutical drugs. Now the studies on in 3 seaweeds and antifungal activity and nanoparticle activities. Thank you. Thank you for this opportunity.
Any questions? So we can move to next presenter.
Okay.
Thank you,
Next one. Next one is P. Kartika Devi with the title Study of Antimicrobial Activity of Leaf and Berry Extract of Physalis minima.
Good afternoon to one and all. I'm Jyotika, third BSc Microbiology, KR College of Arts and Science. My topic is the study of antimicrobial activity of leaf and berries extract of Physalis minima. We done this project under the guidance of Mr. D. Kani sir. The overview of this project is introduction, aim and objective, Physalis minima, isolation of bacterial pathogen, procedure, well diffusion method, thin layer chromatography and result. Now we're going to see the introduction. The many antimicrobial agents are available, but microorganisms show resistance to most of this agent. The plants are known to produce a variety of compounds to protect themselves against a variety of their pathogens. It considered as potential source for different classes of antimicrobial substance. Next, aim and objective. And the present study is to detect the antimicrobial activity of Physalis minima against some gram-positive and gram-negative bacteria. And the antimicrobial activity is done by a well diffusion method and thin layer chromatography method. Now we see a small intro of Physalis minima. It's a small herbaceous perennial annual plant belonging to Solanaceae family. The common name is native gooseberry, wildcap gooseberry, pygamic ground cherry. In Tamil, it is called as Sorakatakali. It grows 20 to 50 centimeter. It forms like a tomato. That fruit is a rich source of vitamin C, and it is used for treating diseases like cancer, leukemia, malaria, asthma, hepatitis, dermatitis, and rheumatism. And it is naturally bitter in taste. Next, isolation of bacterial pathogens. We isolate seven organisms there: Staphylococcus aureus, Pseudomonas, Escherichia coli, Bacillus, Salmonella, Shigella, and Vibrio cholerae. The isolated organisms are confirmed by various biochemical tests like IMBIC, catalase, oxidase, urease, H2S production, carbohydrate fermentation test, nitrate reduction test, and hydrolysis test. These are the selective media plates for isolate the pathogens. Next, procedure for preparation of extract. The plant's leaves were dried and grind into fine powder. The powder were extracted by using the succulate apparatus using the solvent methanol. Then extracts were evaporated to reduce concentration 1/3. Next, fruits were crushed and soaked in methanol and kept in water bath for 1 hour, 1 day, and the solvents were filtered, and the crude extract is used for further purpose. Next, well diffusion method. This method is used to detect the antimicrobial activity. Here we used Mullen-Hinton agar plates. We all know that antimicrobial activity procedure. After incubation, the plates absorb a zone. It will indicate the result. The same procedure is used for berries. Next, thin layer chromatography. It is used to detect the compounds whether it is present in extract or not. Here we used leaf extract. In leaf extract, some compounds are present that are shown in the picture. In a berries extract, there are no compounds are present. At last, by above mentioned method, we conclude that in berries extract, the zones are absorbed only in Vibrio cholerae. In leaf extracts, the maximum zones are absorbed in Staphylococcus aureus, and minimum zones are absorbed in Bacillus. Thank you for giving this opportunity.
Any questions from audience side? Okay, what are the solvents you used, Jyotika? Only methanol?
Yes, ma'am. Only methanol we used.
So, why can't you try for another other solvents like polar or non-polar solvents?
Basically, Soxlate apparatus, we use 3 solvents: methanol, ethanol, benzene. Out of these, we took methanol, ma'am.
Okay. Okay then.
Thank you, ma'am.
Next is Gejalakshmi with the title DNA Vaccines.
Is this clear, ma'am? Hello?
Yes. You are audible and your PPT is visible.
Okay, thank you, ma'am. A very warm good afternoon to all of you. I am Gajalakshmi, studying pre-final year of biochemistry in KR College of Arts and Science. I am here to tell a brief about DNA vaccine. All of you know Edward Jenner in 1798 who discovered a vaccine. Now we see what is DNA vaccine. A vaccine uses a gene from a virus or bacteria or whatever is used to stimulate the immune system. It was made up of a small circular piece of bacterial DNA plasmid. It has been tested for veterinary use only. As of August 2016, no DNA vaccine had been approved in the US for human use. So this is how the vaccines are formed from West Nile virus. The RNA of West Nile virus is extracted. The RNA is converted to DNA by reverse transcription. The DNA represents the West Nile virus genome. The genetic sequence of West Nile virus is generated from the DNA, and this is introduced into the DNA synthesizer. Based on the DNA sequence, primers specific to the primium and E genes regions are produced. These primers will in turn used to generate cDNA fragment containing the E genes. The cDNA fragment is then inserted into a circular piece of DNA called a plasmid. The plasmid carrying the primium and E genes are grown into large quantities in bacteria and purified by column chromatography. The purified plasmids are made up into the investigational vaccines. So then why DNA vaccines are discovered? In 1983, Enzo Pelletti and Dennis Panicali at New York Department of Health devised a strategy to produce DNA vaccine by using genetic engineering to transform ordinary vaccine into vaccine that may able to prevent such a genetic disease and other diseases. It has no risk for infection. It stimulates the ability of humoral and cell-mediated immunity. No refrigeration is required. So then, the mechanism of DNA vaccine. It is firstly the plasmid DNA with antigens are injected into the myocytes. Then it was directly transfection into B cells. The cross result from the transfected cells are phagocytosis by APCs. Then antigen design. DNA vaccines antigen design can direct the post-translational trafficking of protein to influence the development of host immunity. The human tissue plasminogen activator reader promotes high level of protein secretion and improved antibody responses. Then clinical targets. Till now, 43 clinical targets have been done for viral and non-viral diseases. 33% in HIV and 29% in cancer, 38% in influenza, hepatitis C, malaria, etc. DNA vaccine directed at eliciting an immune response towards tumor appearing for both prophylactic and therapeutic treatment for cancer. Completed trials. Phase 1 trials gave more failure due to poorly immunogenic, low antibody T-cell responding. In phase 1, HIV research gave negative results too. The only approved vaccines are FDA and USDA for veterinary use only. Then ongoing trials are improvements in humoral and cellular immune response and activate CTA cell than phase 1 trials. Like HIV, influenza, and others, about cancer is also studied in the research. DNA vaccine to attack cancer. How DNA vaccine attack cancer? Firstly, DNA vaccine is injected to DNA genetic part CpG to produce protein material secreted by specific cells of immune system like cytokines. Then DNA vaccine enter into cancer inducing cell and branches into endogenous and exogenous. Endogenous activates cytotoxic T lymphocytes via NHC1. NHC is a material bind with peptide from pathogens and display on the surface, cell surface. Then exogenous activates B lymphocytes and T lymphocytes via NHC2. It includes both cellular and humoral immunity to prevent cancer by cancer immunotherapy. The advent of improved methods of physical delivery and other technologies has spurred a second wave of trials investigating DNA as standalone. Thank you.
Gajalakshmi, what type of vaccine is our COVID vaccines? Covaxin, Covishield, Sputnik, etc.
Yes.
What type of vaccine? It's a DNA vaccine or attenuated vaccine? Gajalakshmi, can you hear me?
Yes, ma'am. Ma'am?
Yes. What type of vaccine is our COVID-19 vaccine? Covishield, Covaxin, etc.
No, ma'am, but DNA vaccines for COVID are in research, ma'am.
Okay. But this Covaxin and Covishield, both are not a DNA vaccine.
Yes, ma'am.
Some other type, right?
Yes, ma'am.
Can you mention the type?
It's an immunity
or recombinant vaccine. Okay. Any other questions? Okay, we can move to the next presentation.
Thank you, ma'am.
Next, B. Murigaveni with the title Stem Cell Biology.
I'm Murigaveni from Department of Biochemistry, Care College of Arts and Science, Kovilpatti. The human body is the structure of human being. It is composed of many different types of cells that together create tissues and subsequently organ systems. Normal cells or the mature cells are the cells produced by stem cells which are capable of working in a tissue for a specific purpose like digestion, defense, etc. Example, kidney cells, brain cells, etc. This image gives answer for how organ systems form. What is a stem cell? A single cell that can replicate itself or differentiate into many cell types. This is called stem cells. Classification of stem cells. Embryonic stem cells, adult stem cells, induced pluripotent stem cells, stem cell culture, stem cell lines, cord or fuel stem cells. Embryonic stem cells, pluripotent stem cells. Adult stem cells are further divided into multipotent stem cells, oligopotent stem cells, unipotent stem cells. Embryonic stem cells. A blastocyst is a pre-implantation embryo that develops 5 days after fertilization of an egg by a sperm. It contains all the material necessary for the development of complete human being. The blastocyst is a mostly harmless pair of cells that is smaller than the period at the end of this sentence. In its interior is an inner cell mass which is composed of 30 to 34 cells that are referred to by scientists as pluripotent because they can differentiate into all of the cell types of the body. In common use, embryo can refer to all stages of development from fertilization until somewhat ill-defined. When it is called fetus, scientists use terms such as morula and blastocyst to refer to precise basic stages of pre-implantation development. In order to be as precise as possible, this booklet uses scientific terms when describing scientific concept, but uses the term embryo. In normal development, the blastocyst would implant in the wall of uterus to become the embryo and continue developing in a mature organism. Its outer cells would begin to form the placenta, and the inner cell mass begin to differentiate into progressively more specialized cell types of the body. Adult stem cells. Adult stem cells supply new cells as an organism grows and to replace cells that get damaged. Adult stem cells are said to be multipotent, which means they can only change into some cells in the body, not any cells. For example, blood stem cells can only replace the various types of cells in the blood. Skin. Epithelial stem cells provide the different types of cells that make up our skin and hair. Induced pluripotent stem cells. Pluripotent stem cells are scientists in laboratory. Induced means that they are made in lab by taking normal adult cells like skin or blood cells or reprogramming them to become stem cells. Just like embryonic stem cells, they are pluripotent, so they can develop into any cell type. Stem cell culture. Growing cells in a laboratory is known as cell culture. Human embryonic stem cells are generated by transferring cells from the pre-implantation stage embryo into a plastic laboratory culture dish that contains nutrient broth known as culture medium. The cells divide and spread over the surface of the dish. However, if the plated cells survive, divide and multiply enough to crowd the dish, they are removed gently and plated into several fresh culture dishes. The process of replanting or subculturing the cells is repeated many times and for many months. Each cycle for subculturing the cells is referred to as a passage. Once cell line is established, the original cells yield millions of embryonic stem cells. Embryonic stem cells that have proliferate in cell culture for 6 months or more months without differentiating or pluripotent and appear genetically normal are referred to as an embryonic stem cell line. At any stage in this process, batches of cells can be frozen and shipped to other laboratories for further culture and experimentation. Stem cell lines. A stem cell line is a family of constantly dividing cells that product of single parent group of stem cells. They are obtained from human or animal tissues and can replicate for a long period of time in vitro within glass or commonly in the lab in an artificial environment. They are frequently used for research relating to embryonic stem cells or cloning entire organism. Once stem cells have been allotted to divide and propagate into controlled culture, the collection of healthy, dividing and undifferentiated cells is called a stem cell line. Cord blood stem cells and amniotic fluid stem cells. Cord blood stem cells are harvested from the umbilical cord after childbirth. They can be frozen in cell banks for use in future. These cells have been successfully used to treat children with blood cancers such as leukemia and certain genetic blood disorders. Stem cells have also been found in amniotic fluid. This is a fluid that surrounds a developing baby inside the mother's womb. However, more research is needed to help understand the potential uses of amniotic fluid stem cells. Stem cells in medicine. The only stem cells now used to treat diseases are hematopoietic stem cells. These are the blood cell forming adult stem cells found in bone marrow. Every type of blood cell in the bone marrow starts as a stem cell. Stem cells are immature cells that are able to make other blood cells that mature and functions as needed. These cells are used in procedures such as bone marrow transplants. This helps people with cancer make new blood cells after their own hematopoietic stem cells have been killed by radiation therapy and chemotherapy. They may also be used to treat people with conditions such as fanconic anemia. This is a blood disorder that causes the body marrow to fail. Stem cells may help your health in the future in many ways and through many new treatments. Researchers think that stem cells will be used to help create new tissue. For example, one day healthcare providers may be able to treat people with a chronic heart disease. They can do this by growing healthy heart muscle cells in a lab and transplanting them into damaged hearts. Other treatments could be target illnesses such as type 1 diabetic, spinal cord injuries, Alzheimer's disease, and rheumatic and arthritis. New medicines could also be tested on cells made from pluripotent stem cells. This is an image source of bone cells. Need more dedicated on board how to control these cells. Another challenge is that the embryonic stems available today are to be rejected by the body, and some people find it morally troubling to use stem cells that come from embryos. Scientists face challenges when using other pluripotent these these cells are also found in some of the body. There is a great chance that if you are interested in three certain condition, ask your healthcare provider how to find out about people in your area. What are the benefits of stem cells? They are non-toxic and devoid of side effects. Toxic parts. For therapeutic purposes can ensure lower treatment cost and promote longer lives. Stem cell therapy addresses both better treatment and giving longer life and lower cost. This is how many diseases can be cured by key applications of stem cells purposes. Apart from bone marrow transplantation, stem cell therapeutics is still in the developmental stage. Attempts are being made to use stem cells in treating type 1 diabetics, Parkinson's disease, Huntington's. However, is and no applications help after radiotherapy. Skin can be to of a patient. Their hair follicles contain skin stem cells or carotenoids, and these cells can be isolated and cultured to grow an epithelial sheet. This method can help to reduce the need to use skin grafts from other people, lowering the chances of reflection. Brain cell transplantation. Stem cells can be employed to generate dopamine, a chemical that is lacking in patients suffering from Parkinson's disease. However, in some cases, patients develop side effects suggesting the presence of very high levels of very sensational in this method. What is stem cell banking? Stem cell banking is a process of collecting, processing, and storing potentially life-saving stem cells for future use in therapies and regenerative medicine. With their ability to develop into many different types of blood and tissue cells, stem cells have the potential to regenerate and protect the body from within. In cases where a disease or condition affects cell tissue, stem cell therapies can be used to repair damaged tissue, repair organs, and treat diseases. Conclusion. There is a lot of research on investigating how stem cells can be used to treat diseases. It is expected that in the future, stem cell therapies will be developed for many types of diseases for which there are no effective treatment at the moment. Basic studies stimulate to Thank you.
What is the function of hematopoietic stem cell?
Murugavani, what are the importance of stem cells?
This is to cure untreatable diseases, ma'am.
Okay. Yeah. Yes, I heard. Any other questions? Okay, thank you. And we can move to the next session.
By this, we end our paper presentation session. And now I invite Dr. R. Shabir Ruksin, Assistant Professor, Department of Allied Health Science, NISHI, to present the conference report.
Good evening to all. I am Dr. Shabir Ruskin, here to present the conference report. The one-day virtual international conference on genetic technology is held on 28 May 2021. The inaugural section of the international conference began with the invocation to Almighty by Ms. Surya S. Krishnan, BSc Human Genetics and Molecular Biology, Allied Health Science, Niche. Dr. G. Arokya Selva Saroja, Head Department of Allied Health Sciences, Niche, has delivered the cordial welcome address. The Honorable Registrar, Dr. P. Thirumal Valavan, Noorul Islam Center for Higher Education, has given the inaugural address, and Honorable Pro Vice Chancellor Niche has given the presidential address. Finally, the inaugural session was concluded with vote of thanks by Dr. J. Rajabrinda, Assistant Professor, Allied Health Science, Niche. The conference was carried out through first technical session. Ms. T. Subha, Assistant Professor, Department of Allied Health Science, Niche, introduced the resource person, Dr. Balasundari Ramesh, Health Service Officer, TX Wing, USA. Dr. Balasundari Ramesh delivered talk on topic Biomimicry and Hematopoietic Stem Cells Blood Factory. She described and explained about the in vitro culture of human RBCs from bone marrow hematopoietic progenitors and also pointed various applications related to this topic. The students will be highly benefited by her presentation. At the end of her presentation, students quoted many questions and the session ended with an interactive session. The conference was followed by Technical Session 2. Ms. G. Monique Lega, Assistant Professor, Allied Health Science, Niche, introduced the resource person, Dr. Phone Lee Queen, Deputy Dean, Faculty of Science, Department of Allied Health Science, University Tunku Abdul Rahman, Malaysia. Dr. Phone Lee Queen shared his in-depth knowledge on polyvinyl chloride microplastics, induce oxidative stress, and pro-inflammatory responses in ICR mice. The talk is more informative and research-oriented. She explained about the serious cause due to the accumulation of microplastics in wildlife and humans. Also, she explained about the disorders caused due to the two defective gene PPARY and CD36 due to polyvinyl chloride, which is a microplastic. Her presentation and dynamic interaction was deeply appreciated. In the technical session of the conference, Dr. J. Rajabrinda, Assistant Professor, and Ms. Sarojini, Assistant Professor, AHS Department, Niche, acted as a chairperson. The conference was attended by more than 380 participants from 78 various universities and colleges all over India, and total of 25 technical papers on various topics such as cancer genetics, stem cell therapy, drug therapy, nanotechnology were presented by the authors. Overall, the conference was really a nice one, truly well organized, maintaining time schedule, and especially with a very nominal registration charge to encourage huge student participation, to enhance awareness for research to meet the need of society. Thank you, one and all.
Now, I would like to invite Dr. NCJ Pakilakshmi, Assistant Professor, Department of Allied Health Science, Nishi, for a vote of thanks.
Good evening, all. I deem it a great honor to propose the vote of thanks in this occasion. I extend my sincere thanks to the resource persons, Dr. Balasundari Ramesh, Second Lieutenant Health Service Officer, US Civil, and Dr. Phone Lee Queen, Deputy Director, R&D, Postgraduate Research, Faculty of Science, Allied Health Science, Uttar, Malaysia, for their valuable information they shared with us. I must express my deep sense of appreciation for them. I would like to extend a very heartiest thanks to our Chancellor, Dr. A.P. Majid Khan, for his inspiration and support to organize this conference. I express my thanks to our Pro-Chancellors, Mr. M.S. Faisal Khan, Dr. R. Perumal Sami, Vice Chancellor Dr. A.K. Kumaraguru, Pro Vice Chancellor Dr. N. Chandrasekhar, and Registrar Dr. P. Thirumal Valavan for their motivation and moral support to organize this conference. I also wish to express my thanks to Department Dr. G. Arokya Silva Saroja and our program coordinator Mrs. S. Arya Sankari for their constant support. I extend my thanks to coordinators, my colleagues for their support throughout the conduct of this conference. I wish to thank Dr. Janaki MC for her technical support to conduct this conference in a successful way. And also, I express my gratitude to Nihala Siddiq and Surya S. Krishnan, BSc Human Genetics and Molecular Biology, for their wonderful Master of Ceremony. Last but not least, I express my sincere thanks to all the students of Allied Health Sciences Department, NICHI, and participants from various institutions for their support. Once again, I express my heartiest thanks to all. Thank you all.
Thank you, ma'am. As we have reached the final phase of our conference, let us conclude with the national anthem.
जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे।
Jani ma'am, post the feedback link in the chat box.
Yes ma'am, it is being posted.
Okay ma'am.